It ain't easy down here in the wild, wild west of the intestinal interstate. But on occasion we get a visit. Oh man, I freaking love this show. How does the immune system distinguish between the law-abiding citizens of the microbiome and the troublemakers? This is a question that I myself have long pondered. While everybody learns that it's the job of the immune system to neutralize all threats, foreign and domestic, how exactly does it do that without taking down the cells that make you, you? And I'm not just talking about the cells that share your DNA, but the innumerable residents of the human microbiome. The vibrant communities of bacteria, fungi, protists, archaea, and viruses that live on the skin, the mouth, the gut, and other places. As with many things at the scale of the little guys, the answer here must be molecular in mechanistic origin. Your immune system doesn't have eyes, nor does it have access to Wikipedia, and yet, for the most part, it manages to keep you in a state that is compatible with life. And the dance that your immune system does to keep it that way is fascinating. But before we get into the specifics, let's go see what's on the TV, shall we? We will return to Cells of Our Lives right after a word from today's sponsor, Brilliant. Brilliant is where you learn by doing, with thousands of interactive lessons in math, data, programming, and AI. Active learning is the best way to learn, and Brilliant's platform is a shining example of making good on that strategy, with lesson designs crafted to make you think and problem solve. My favorite feature of Brilliant is the fact that they make it easy for you to learn on the go, with a mobile app that will allow you to get that little bit of extra learning in between your daily doom scroll. A tool every good scientist needs is data analysis. The course starts off with basic fundamentals that anybody can get into and progresses into practicing analysis with real datasets from Airbnb, Spotify, Starbucks, and more. To try everything Brilliant has to offer for free for a full 30 days, visit brilliant.org/findtheneutrophil or scan the QR code on screen, or you can click the link in the description. You'll also get 20% off an annual premium subscription for using my link. Thank you to Brilliant for sponsoring this episode. And now back to the Cells of Our Lives. It was down by the duodenum where I first laid my eyes on you, T. Regina, dressed to the nines with receptors so sharp they could kill. The immune system is broadly divided into two divisions. These divisions are the innate immune system and the adaptive immune system. The Department of Innate Immunity is the department that writes my checks, with me being a neutrophil and all. It is much older than the adaptive immune system and has evolved to fight off threats non-specifically. Encoded in the genome of every cell of the innate immune system are the instructions to make cell surface receptors that recognize pathogen-associated molecular patterns, or PAMPs for short. The immune system evolved to recognize bits of molecules that pathogens generally like making. This class of molecules is extremely broad, including things like snippets of viral DNA, pieces of a yeast cell wall, and bits and pieces of bacterial flagella. Some PAMP recognizing receptors trigger the release of chemoattractants, which recruit cells like neutrophils to the scene of the crime. Other PAMP recognizing receptors trigger the process of phagocytosis, or cell consumption. PAMPs and the receptors that recognize them play a key role in the innate immune system to detect and fight pathogen infection. The issue here is that the name PAMPs is misleading. Plenty of commensal microbial species also make PAMPs, meaning that PAMP detection is not sufficient to discriminate between friend and foe. Some scientists advocate for a more inclusive name for these molecules, microbial-associated molecular patterns. To summarize, the innate immune system isn't very careful in distinguishing between good microbes and bad microbes. We learned what molecules bad microbes make, which is not exactly a group of molecules that excludes the molecules that good microbes make, leading to potential crossfire. If the innate immune system isn't so discriminating, what about the adaptive immune system? The adaptive immune system is the newest addition to the armies of immunity, and they come packing heat with all the latest high-tech weaponry evolution has to offer. Adaptive immunity, unlike innate immunity, does not rely on recognizing a set of genetically inherited recognition software, but rather relies on the ability to learn to recognize what is considered foreign and what is considered self. If this is the only distinction, how does the adaptive immune system know what's considered foreign and friendly, foreign and unfriendly, and domestic? To answer this question, let's learn a bit about how the adaptive immune system functions. At the heart of the adaptive immune system lie the T and B cells, both of which rely on specifically identifying new threats by making proteins that bind them. But if all of your cells contain the same DNA, and if your DNA codes for protein, How does your adaptive immune system generate a new protein to learn to recognize new threats? Unlike the majority of your other cell types, T and B cells have the capability to scramble a crucial part of their own DNA to randomly generate a novel protein. For the T cell, this protein is the T cell receptor, the cell surface protein that the T cell uses to detect whether a cell is infected or not. A new threat to the immune system will make proteins the immune system has never seen before. T cells, by randomly scrambling their own DNA, can try out trillions and trillions of different receptors to try and find one that can bind to proteins that the new threat makes. For the B cell, this protein is the antibody. This scrambling process is called VDJ recombination and allows for the T and B cells to generate a truly stunning array of protein diversity, up to 3 × 10 to the 11th unique proteins. To put it in perspective, this is roughly the number of stars in our Milky Way galaxy. Not all T and B cells are created equal, however, because if your T and B cells learn to recognize your cells instead of the invaders, it can cause you to develop an autoimmune reaction. So T and B cells learn to tolerate host cells through a process called negative selection. For the T cell, this happens in the thymus. Along the process of T cell puberty comes a pivotal moment where the epithelial cells of the thalamus show the T cell a dizzying array of host proteins. If the T cell reacts to being shown a host protein, the cell death pathway known as apoptosis is activated, deleting that particular T cell. Only T cells that have a functioning T cell receptor and do not strongly bind host protein are allowed to move to a secondary lymphoid organ where they lie dormant as mature, naive T cells. The process by which the B cell learns not to recognize the host proteins is very similar, except that it happens in the bone marrow. B for bone marrow. B. This system is pretty cool and kind of separates T and B lymphocytes as being super special snowflakes coding for a set of very unique proteins, while at the same time preventing these deadly agents from turning on the cells that they were evolved to protect. But that still doesn't really address the titular question. All bacteria, all fungi and protists, friendly or not, they're all non-self. So why doesn't the adaptive immune system just drop the hammer and dispense some indiscriminate justice?
The thing is, Macrophage, I could never love you the same way you love me.
But why, T. Regina?
Well, because you're neither subscribed to Father Neutrophil, nor have you joined his YouTube membership. How else will you get updates on cell and molecular biology deep dives?
I can change, T. Regina, I swear. We've already heard about how the immune system recognizes invaders, but actually the microbial wars start before the immune system is even involved. Surprisingly, a decent chunk of the anti-pathogenic defense system is out of our hands. The microbiome is self-regulating, taking much of the burden off of the immune system. For the rest of this video, we'll be primarily focusing on the gut microbiome. Even before the immune system gets involved with fighting pathogens, pathogens have to survive the gauntlet. And by that I mean the diverse environments that lie between the outside and inside world. Think about how tough that would be for a tiny guy like a bacteria to make work. To be able to be well adapted not only for whatever vector or fomite carries the bacteria, but also to survive the environment of the mouth, the throat, and the esophagus, each of which have unique local features. Then the pathogen has to survive the hot acid pit known as the stomach, and past that, they have to survive your microbiome. If you didn't already love your microbiome, check this out. Despite these little guys not sharing any of your DNA, your gut microbiome actively resists colonization by pathogenic species, protecting the host. Healthy gut bacteria can fight off pathogens in several different ways, including nutrient competition. Commensal E. coli compete for the same foods as pathogenic E. coli, meaning that even if pathogenic E. coli were to enter your body, there's a decent chance that they just starve before getting anywhere. Environmental regulation. Healthy vaginal flora produce lactic acid, shutting out pathogenic microbes that can't thrive at a low pH. This one's my favorite, antagonistic inhibition. Some commensal bacteria in the gut make molecules that kill pathogenic bacteria. What is absolutely crazy about antagonistic inhibition is that some bacteria of the same family will inhibit their pathogenic cousins. E. coli Nisla 1917 makes microcins. Microcins are small toxic peptides that inhibit the growth of related bacterial strains. Microcin E492 is modified by sticking a siderophore group at the end. Siderophores are iron scavenging compounds. To the pathogenic E. coli, it thinks it's going to get some much needed iron when in reality E492 sneaks in past the outer membrane, where it can then bind the inner membrane protein mannose permease, putting it in the perfect position to pop a hole in the membrane, making sure that the offending pathogen can't keep its insides in and its outsides out. Not only do your friendly gut microbes stifle pathogens from colonizing the gut, your microbial defense force can actually blunt the ability for pathogens to cause disease. Saccharomyces boulderi attaches to salmonella, preventing them from getting to the liver. Bifidobacteria make acetate, which helps the epithelial cells react and block the Shiga toxin from getting into the bloodstream. See my Archaea video if you want to know more about that. But to me, what's most surprising is that the microbial defense force includes bacteriophage. These bacteria-infecting viruses lie in wait in the mucus layer that surrounds your intestinal cells like ambush predators. Bacterial pathogens that like to bury into the host cells must traverse this mucus membrane through a field of bacteriophage landmines. And then and only then will they be granted the opportunity to infect you. It's a little cheesy to say that your microbiome loves you and wants to protect you, cause the real biology nerds in the chat will roast me for doing so. We're all just walking assemblages of organic material at the end of the day, and love is a human-defined concept. Instead, what really gets to me emotionally is the fact that you didn't have to ask your microbiome to do this. It does this because it needs to do this to survive. Since our early beginnings as a meat tube, and that is the last time I will say meat tube, our ancient ancestors found a way to not only survive, but thrive through the terrible storm known as existence. You might not remember it, but you have weathered apocalyptic extinction after apocalyptic extinction. Through plagues and famine, through brutal competition and desperate violence, you and your microbiome are still here today. It isn't love, it's deeper. It's mutual dependence that only survivors know. And only through eons of tragedy do we get endless forms, most beautiful and most wonderful, being evolved. And I just think that's kind of neat.
After all these years, the home that we built, what am I supposed to tell little progenitor Pete?
It was nothing personal, Marcophage. You practically invited me into your life, and I merely accepted. And it ain't T. Regina, it's Typhimurium. S.typhimurium.
We're gonna get to the title question, believe me, just stick with me for a bit longer, okay? Before we talk about how the immune system hunts bad bacteria, we first have to talk about the last line of defense separating pathogens from your innards. If a pathogen miraculously gets past the varied and deadly environments of the mouth, esophagus, and stomach, as well as fending off the gauntlet of friendly microbial guards, colonization and infection then become possible. But it won't be easy. Two layers separate microbes from your vulnerable bloodstream: the mucosa and the intestinal epithelium. The walls of your GI tract are stapled shut by these packed cells connected with tight junction proteins, keeping invaders out of circulation. But not only that, the epithelial cells are coated in a mucosal layer. Here's something that I didn't know prior to doing research for this video. The mucosa are actually an insane tool for the immune system. Mucus is a complex hydrogel that is made of sugars linked to proteins known as glycoproteins. This viscous layer is not only difficult for pathogens to swim through, but also contain a bevy of antimicrobial compounds that stop threats before they make it to you proper. Commensal bacteria are often happy just sitting on top of the mucus layer, eating away at the rich sugar that it provides. But for those pathogenic bacteria that want to get away from the harsh environment of the gut to start colonization, they have to make it through that layer. Many of these pathogenic GI bacteria need to tunnel through the mucus, and they do so with their flagella. Bacteria like Helicobacter pylori evolved spiral-like bodies and flagella to dig through the mucous membrane. But the host is just as clever. The mucus layer is heavy with an immune surveillance protein called TLR5 that binds specifically to bacterial flagellum. This surveillance protein recruits and alerts the immune system when the mucus layer has been breached by pathogenic ne'er-do-wells. Innate immune cells like neutrophils can be deployed within this thin layer to neutralize the problem. In other cases, antibodies that opsonize and body block bacteria and toxins can also be secreted from the gut lining to fight off pathogens from a distance. But here's the coolest thing I've learned about mucus. Mucus can actually tame bacteria and coax them out of a pathogenic lifestyle. Pseudomonas aeruginosa, according to the World Health Organization, is one of the leading threats on human health through evolving antibiotic resistance. However, P. aeruginosa is considered only an opportunistic pathogen. becoming dangerous only when the conditions are right. In a healthy human, our mucus actually causes P. aeruginosa to give up a life of crime and kind of hang out in a planktonic stage instead of forming a nasty, disease-causing biofilm. This is mediated through the protein mucin, which downregulates the expression of P. aeruginosa's virulence genes. This surveillance of the health of the mucus layer is really important to the titular question because whether the immune system fights or not is dependent on context. See, in times of peace, your microbiome actually trains the immune system to relax. Dendritic cells pass off signals to macrophages and regulatory T cells that promote an anti-inflammatory, immunotolerant environment. But through immune sensing, through sensors like TLR5, the immune system can flip from immunotolerant to ready for war. Especially if the damage is so severe that your gut wall breaches, the immune system has to be ready to stop the flood of foreign microbes, commensal or pathogenic. To quote a Nature review paper on this matter, figuratively, this situation is a dangerous armed peace in the manner of the advice, if you seek peace, prepare for war. So we are finally able to put it all together, an answer to this deceptively complex question. How does the immune system tell between good bacteria and bad bacteria? The trick answer here is, the immune system kind of doesn't have to. Sorry for baiting you. Any antigen not tolerated by the immune system is recognized as non-self. So commensal bacteria will still trigger an immune response, no matter how cute and friendly they end up being. This is balanced by the fact that commensals release signaling factors that tame the immune system, shifting the balance to immunotolerant. A pathogen upsets this balance in various ways by alerting the immune system to the telltale signs of infection. A buildup of toxic protein can trigger immune sensors. Damaged and dying cells can send signals of distress. Over time, these signals override the pacifism signals from the healthy microbiome, and you get a proper immune response. Many pathogenic infections localize to certain areas of the GI tract, and signaling directs the immune system to the site of infection. As long as there are signals of damage and distress, these immune cells will do battle until the drums of war cease and immunotolerance is re-established. And if harmless commensal bacteria end up in the crossfire, those are considered acceptable losses. The answer to this question was both surprising and extremely unsurprising at the same time. But what still bothers me is that this system is only perfect on paper. We know how clever pathogens can be. What happens when the battle goes off script? Helicobacter pylori is a triple S-tier pathogen. It's estimated that over half of the human population hosts this nasty bug. H. pylori invasion, if left unchecked and untreated, can develop into a lifelong infection. Infection can cause painful ulcers, and over time, H. pylori infection can cause various cancers, putting it in the horrifying category of carcinogenic infection. All the more reason why research into fighting back this bug is so valuable. H. pylori is ferociously dedicated to messing your day up. Despite being a neutrophil, not a neutrophil, H. pylori is actually able to survive the harsh pH of the stomach by pumping out an insane amount of urease. Urease is an enzyme that converts urea into ammonia, which is a base that can neutralize acids. Urease is extremely important for H. pylori, and there's some evidence that shows that leftover urease from the corpses of dead H. pylori are taken up by the survivors in a process called altruistic lysis. Although, as I was perusing the literature, it seems like there's a bit of disagreement if that's technically true. Once H. pylori settles on an area in the GI tract to colonize, it beelines for the mucosa. H. pylori evolved to flourish in the mucosal niche, and if you think about it, that's a really smart strategy. On one side of the mucosa, cutthroat competition, and on the other, the armaments of the immune system. The mucosa provides a relatively peaceful place for H. pylori to remain unbothered, moisturized, happy, in their lane, focused, and flourishing. Part of the immune evasion mechanisms that H. pylori uses is impersonating commensal-friendly bacteria, tricking the dendritic cells into lowering the defenses of the immune system. This shifts the balance of immunity to tolerance rather than combat. H. pylori is full of other nasty tricks. Maybe we'll get into some of those in a Phibit or a Journal Club stream. But with the immune system dampened on one end, and with the protection of the mucosa on the other, wiping out an H. pylori infection without the use of antibiotics is a major uphill battle, thanks to the unique immunological architecture of the gut. I started the idea for this video very genuinely. I had no idea how the immune system distinguished between good and bad microbes. I knew that little guys like me were involved in our innate immunity, our body's natural ability to recognize and respond to markers of pathogenicity, but I had no idea that the natural architecture and interface with the microbiome itself had so many defenses that resist infection even before the immune system is recruited to battle. It's also given me a new appreciation for our ride or die relationship with our microbial friends. If it's any consolation for you out there, you aren't alone. You have like 40 trillion cells who are ready and willing to shank their evil cousins for you. Thank you to my YouTube members for supporting this channel, it really means the world to me. If you've liked this video, why not subscribe for more cell and molecular biology content?